Great. Good evening, everyone. I feel like this talk really takes what we just looked at, where David was kind of describing association studies. We've accumulated studies. The real kind of question is can we move towards a more structured representation of this data that would enable sort of biologists to quickly kind of come to hypothesis or easily testable hypothesis in the lab. So, like I said, we have all this interesting genotype and phenotype data, and we've been able to account for such little % of the inheritance. And so the question is can we look sort of further into the data and be able to come up with a quick bioinformatic way where we can extract knowledge from these and quickly move to biological experiments that we can then partake in. So today I'm going to be focusing on particularly epistatic interactions or gene interactions that you can extract both from as well as other approaches. So I'm going to give a little back to kind of two general approaches that people have taken to sort of uncovering gene epistatic interactions. So the first is based off of sort of these approaches, mapping gene interactions, and these are things like linkage studies or eQTL studies. And this kind of study usually start with a particular population that already has some continuous variation in a particular phenotype, and you sort of map that to a particular sequence variation population. So do this in terms of pairwise marker association. So the goal is to essentially find marker pairs that jointly explain the variation in a phenotype better than either marker type alone. So there's a small example here. Imagine we had some haploid organism in this case, and we're looking at two different markers, marker B and marker C. And what we see is that if we partition the population based on whichever particular allele they have at these two loci, we see that neither loci alone accounts or can explain the variation seen in that given quantitative phenotype. However, if we move to sort of a pairwise or a joint view of that, we notice the alleles at loci B and loci C can jointly sort of partition phenotype. And that's because that suggests that there's some interesting gene between markers, and this may yield new functional or in between genes. Brings with it sort of a whole host of issues, to sort of not really knowing what the causal gene here is that things have been with a lot of issues of trying to garner statistical significance. Well, it's interesting, but there's been all kind of interactions, and we thought, can we turn to this field and apply some of the learning? So, in sort of the yeast community, especially, been able to rapidly generate these gene-gene interactions using Rohith. So, start with a population of interest. Yeast genetics and elite pairs of genes in different strains, and then measure what that outcome is in a particular phenotype. So in particular, Charlie Boone lab at the University of Toronto came up with this synthetic genetic array, and I won't go into too much detail, but essentially you can engineer different mutations shown here in this gene to the left and the right here. A series of mating and relation steps, you can finally arrive at a double mutation, and typically people will use array these in very high density arrays, and essentially one versus 1536 or 384, and you can take a picture and see offer much smaller. Then you can sort of action between the genome. This might be the wild gene alone result in reduction in the colony size, and then based on that, some expectation of genes had any deviation action where the colony would grow much slower, or a positive interaction. So this community been able to do genetic interactions in the Cerevisiae community have been enormously useful in sort of defining pathways, concepts of pathways, and interrelationships with pathways. And a lot of these interpretations is from a really famous paper by Avery and Wasserman nearly 2 decades ago. So what they came up with was with simple explanations to explain either a positive or a negative genetic interaction. So in this example here, we have two pathways. One is just a simple straight linear pathway leading to some product Z. And in that case, we have two parallel pathways which lead to some other product P, which is also, let's say, essential for cell growth. And so if we knock out sort of gene X and gene A, we've sort of perturbed two completely separate different pathways, and we wouldn't expect any sort of, so this would be the definition of a neutral interaction. These two pathways have nothing to do with each other, and so if you knock out pairs from either pathway alone, you really won't get much deviation from sort of the neutral model. However, if we knock out sort of gene A and N, which are components of these parallel pathways, the disruption, there's no more redundancy here, right? And so, this leads to sort of a loss of this statistical product, which would then manifest as a negative genetic interaction. Positive interactions, on the other hand, sort of represent perturbations to a single or the same pathway. And the logic here is that, if you've knocked out one gene, you've sort of already removed that pathway, and any further perturbations to that particular pathway is not great as, perturbing a completely different pathway. And so you would expect this cell to sort of grow better than what you would expect. So taking this, our group and other groups have been able to sort of formalize this logic to automate the discovery of both within pathways or complexes and these between pathways that I was thinking of. And so the sort of the two models that we've come up with are these within complexes, which define as sets of proteins that are densely connected by physical interactions, as well as positive or negative interactions. And these are usually these models are usually sets of proteins that have a really coherent biological function, like a protein complex or signaling pathway. On the other hand, we also have this between complex model, which we define as basically sort of two within complex models, which are now scanned by sort of a dense bundle of genetic interactions. And the key feature here is there lack physical interactions between these two things. And so what this sort of represents are sort of synergistic or functionally redundant pathways. And we come up with algorithms to sort of automate this process and essentially prepare genes for generations to global maps of each node here is a protein complex. Here is protein complex connected to protein interactions, and then genetic interactions of genes to formulate hypotheses. Given all this genetic interactions derived similar, can we move from complexes? So yeah, like I said, so our goals in this study are basically to ask, can we use these to be what we termed a natural genetic network? To what extent can this network reveal, novel cellular pathways? And then another interesting question since we're working in yeast is how well does this network, either from genome-wide association or linkage studies, how well does it correspond with synthetic networks where you manually perturb pairs of genes? To do this, we used a cross which was done in yeast, and what they did was they made the wild strain was a laboratory strain and a raw strain. Each of these strains was genotyped at roughly 16 chromosomes, simultaneously also being phenotyped thousands of gene expression, the genome-wide expression profile. The question for that step to do is find a way to detect marker to those details in the paper. We use a stepwise scale rather than markers joint association with trait. Detected and simply scanned all other marker phenotypes. But even with that, doing that sort of shrinks your search space, which sort of improves your type 1 error. But even after doing that, what we discovered was there were only about, in that study, 126 significant marker-marker interactions. So we thought, how can we improve this right off the bat? And so the first thing we did was to simply we came up with a pretty interesting observation. And so what we noticed so what you're looking at here is these are segments of different chromosomes. Black bars represent markers that were genotyped in this study, and a solid line between the two are significant marker-marker interactions that were found in this study. And so what we discovered was that often what the case is that because of contiguous markers will typically be linked due to a phenomenon where there's just few recombinant contiguous markers, which basically means that markers that are right next to one another are essentially redundant. And so if we find what we found was if we find one marker, we typically in the markers that are immediately adjacent to it will get patterns of marker interactions. So it sort of looks like essentially two are connected by a bundle of marker interactions. And so we thought, how people utilize the HapMap project, where they will take haplotype and select a single tag snip. We thought, can we turn this sort of on the head, and instead of selecting tag snips, because that sort of requires either threshold as what are linked markers, we thought, well, instead, can we just sort of go through the list, take in more marker interactions that may not have garnered genome significance, and sort of come up with a way to learn from that, transform it from marker interactions, interval interactions that are made by many marker interactions. So what do I mean by this? So let's just imagine. So this figure, panel B over here, is sort of the same view that you're here, but we've transformed to sort of a matrix view. Okay. So here, each row or column represents a particular marker. In this case, on chromosome 13 and chromosome 15, and each sort of individual small box here represents one of these marker interactions. And the blue ones have been coded blue, green ones have been coded green here. And so we thought the goal of this sort of our idea is can we try to define interval interactions represented here by rectangular boxes that contain a significant or higher than expected number of marker interactions. And we thought that might have sort of two beneficial properties. Okay, the first is that, like I mentioned, we have the list of marker interactions, ones that necessarily might not have garnered, but we can allow several of these marker single interval interaction. So each of these alone may have been p-values, but together you would have not, you wouldn't have configuration. The second thing is a lot of us, Genome-Wide association studies, suffers from this map problem. We are just identifying stretches or intervals of DNA, but this interval could contain, how do you map marker favorite method? You might say, look at all genes up to base pairs, but that's kind of arbitrary. And so we thought, by a strict interval, we can sort of narrow down, or honestly, the interval of DNA underlying, the variance in this case. Okay, I have this interval here. I'm going to look only at genes this interval or contained. So it provides from move between marker interval to genome mass. Essentially, this is a biclustering where biclustering these rows and columns, and so we designed an algorithm to exhaustively look at all possible rectangles. Okay, every single possible rectangle up to a maximum size of about, and so we were able to transform 4,687 mark 2,000 pairs. So now we have sort of a natural interval network, and to ask really biological questions. Can we elucidate complex, and how similar is this network to synthetic? Okay? So the thing we have to do, like I mentioned, is we need a mapping scheme to move from intervals to sort of gene pairs, right? And so, like I said, this approach offers us the benefit of being able to sort of logically map these things. So in this case, let's say this interval is found to interact with this interval. Gene A, let's say, is overlapping or contained with this interval. Genes B and C are overlapping or contained within this interval, okay? And so what we would say is this interval-interval interactions supports gene pairs AB and AC, okay? And so now we have sort of a mapping from interval-interval interactions to gene-gene interactions, and now we can begin to ask all those interesting questions. But now some of you might be thinking, there's somewhat of a problem here, okay? Notice that each interval is obviously identifying contiguous genes, okay? And if we want to explore, do any sort of functional analysis, we know that gene order is not random along the chromosome, right? Contiguous genes have been known to be co-expressed. Contiguous genes have been known to co-functionalize with one another. And so we run into certain problems if we're trying to identify, complex complexes that are spanned by a significant number of gene-gene interactions. And the problem is this. Let's suppose we have some complex, let's call it complex red, which contains 3 genes here, denoted by these 3 circles, and all 3 of those genes, let's say, are, one after another a particular chromosome, okay? Similarly, we have this purple complex, which also has 3 genes, and again, they're all located one after another on this chromosome. What unfortunately happened using our scheme here is that this one interval-interval interaction will essentially identify all pairs of interactions between these genes, and you would then say this is a significant complex interaction. But that's not appropriate. I mean, we find it as an artifact or sort of a limitation of the data. So what we thought, we thought about this for a while, and so what we came up with is for each complex or functional term that we're kind of going to search for enrichments, we assigned a particular score, and I won't go too much into the details, but essentially this score spans a range from 0 to 1, which essentially measures the degree to which the member genes of this complex or functional annotation are either clustered, in which case p-min is equal to so if the genes are very tightly clustered, I apologize, the typo here, the p-min will have a value of 0, and if they're completely dispersed along the genome, then they'll have a value of 1, okay? Essentially, this is a probability of measuring how close together these genes are compared to sort of random sets of genes that you could pick, right? And so how do we calibrate this parameter? So what we did was we came up with a permutation scheme. So oftentimes in genome-wide association studies, people will basically permute the phenotype label that an individual will have, thus breaking the connection between genotype and phenotype, while simultaneously maintaining any linkage structure that we see in the markers, right? And so we went back to the original data, we permuted the phenotype labels, we recompute like 1 million marker networks, bi-clustered those, and then asked how many significant complex interactions do each of these random networks go. And we plotted that as a function of p-min, okay? So thresholding our list of complexes at 0, 0.1, 0.2, 0.3, etc. We then plotted sort of the number of complex interactions or between pathway models that our network identified. And as is the case, you'll notice that if we sort of allow all complexes, this problem definitely arises, and even random networks will identify several significant between pathway modules. But if we start to increase this p-min, we see this number falls. And so we picked a value of 0.1, which results in something like less than 1 complex interaction, significant complex interaction identified by a randomized network. And we did a lot of other different permutation schemes and essentially showed that by basically thresholding our list of complexes based on this metric, the number of false discoveries that we allow in due to this phenomenon is relatively low. Okay, so what is the result? The result is we find these sort of beautiful between complexes. So in this case here, we have different members of the synaptonymic. Black lines represent physical interaction between the nodes. And then race 2, and again, each node, different gene, black lines represent between them. And so what we're noticing here is that basically each of these particular genes, which are being found to interact with particular genes, and immediately sort of both boosts sort of these genetic interactions that we're finding, as well as sort of improving the power, because given one of these sort of marker-marker interactions harbored in each one of these genes in and of themselves, you combine them sort of logically in this, the probability this, and this, and so it boosts that power, and in addition, to come up with hypotheses that we then go in and test. So, for example, a simple one is how the aptonemal complex is buffering or is synergistic, right, to forgiven function. Okay, and just bypassing sort of definitely improve the number able to find. Okay, so to it, we can then generate global module. Okay, and each link between. What's cool about this, which synthetic lethal data, is 5,000 is linked to or control. What we thought gene expression traits and their function, and the functional enrichment. So, roughly 2/3 of the are highly functionally existent for marker pairs, suggesting that by and sort of definitely. Maybe I'll gloss over this. Overlap with us being pairs of genes and different publication, genetic interactions between particular subsets of genes. And what we found is the lab is actually rather small. Somewhere around 8 to 9% of these networks contains gene-gene interactions that were also found in our network. However, through permutations, this, although it's a small fraction, it's definitely a significant fraction. Okay, and so there's a lot of different reasons, and the paper I'm going to leave you at one being, there's sort of this GWAS versus Genome-Wide. It's much harder to do, but on the other hand, GWAS by the number of samples that we've collected. And so is that you'll see the gene expression, whereas each of these syntheses, so it's kind of obvious. You wouldn't really, and so but there actually is a specific overlap, and so we sort of investigated in detail and found sort of the gene interactions for common both of these next were particularly core sort of is complex if that works. So these are as well as natural selection. And again, you'll see a causal again both an impact on both. So really interesting thing here is like I mentioned, so when we're at the higher genome in one, some lab tech has to go in. And so we thought, well, there is a small space that is complementing the natural genetic interaction, synthetic interaction. Okay. So I should note here the dimensions of this complex has in the natural network, notice all of these complexes or boxes are interacts supported by the natural network. The reason that is very apparent. So here, each point represents sort of the significance of a complex interaction, either in the SG synthetic network versus natural network. And what you notice here is that there's a huge complement at a p-value 1, reflecting the fact that it's not that they weren't found in synthetic interactions, but really that they just hadn't been tested. And so to cut the story, we tested a bunch of interactions we'd found in the natural network, genetic, and we're able to sort of recapture or successfully recapitulate complex interactions that we found in the natural network. I'd like to sort of thank my co-author, co-author on this paper, my PI, and the numerous that make it possible. Thank you very much.
Mm-hmm. We talked about the effect of the power of the effect of the power of the test has based on the genome-wide association studies on these samples.
Right.
Comment on that?
Yeah. So the question was reflecting on sort of. So I mentioned that one of the differences between the two types of networks was sort of the power to detect a single gene-gene interaction, and so. Basically, although genome-wide association studies have sort of unbiased, sort of unrivaled sort of coverage in terms of looking at all portions of the genome, what we're essentially looking for are correlations, right? We're trying to link already present perturbations in the genome to a given phenotype or trait of interest. And so that involves, correlation tests, statistical tests. And so the power to detect a given effect is limited by how many individuals you've assayed for both genotype as well as phenotype. And so a lot of people have done simulations and basically showed that obviously as you have more people, you have more power to detect a given gene-gene interaction. This simply isn't a problem in sort of synthetic lethal screens because you're going in there and manually in an isogenic strain perturbing two given genes, right? And you're completely knocking them out, completely gone. And so there's a lot more power to detect a given change there.